Hi, everyone. It's good to see you all. Welcome to our January 24th EMSL Learn webinar on 3D imaging platforms for live cell microscopy. So thank you so much for joining us today. We would love to hear from where in the United States you're joining us from. So if you can go ahead and drop in the links, your location, that would be fantastic. We are, us EMSL folks are here in Richland, Washington. Our next EMSL Learn webinar is going to be on February 7th, and it's going to focus on using EMSL metabolomics technology to advance your research. The best way to keep in touch with EMSL and to learn about all of our upcoming virtual, hybrid, or in-person events is to make sure you subscribe to our email list, and also keep in touch on the website. We have a lot posted there, and also on our social media channels on many different platforms. I mean, pick one and we're probably there. So for today, as a speaker, EMSL offers access to live cell imaging platforms for your biological research. And today, you're going to hear from EMSL biologist Erin Bredeweg as she talks through EMSL's latest platform called NanoLive. And you'll also hear from Dehong as he discusses EMSL's Lattice light sheet capability for 3D imaging. Both speakers are going to go over how these platforms can be used, their strengths and limitations, as well as sample requirements. There will be time at the end of today's webinar for questions. And so we encourage you to post your questions in the Zoom chat so that Erin and Dehong can answer them during our Q&A. And so with that, we will begin with Erin. Take it away, Erin. Erin, are you there? Sorry, folks, it seems like we might be having some technical difficulties. Erin, can you put yourself on camera? Sorry, everyone. Yeah, we seem to be experiencing some technical difficulties here. We are trying to quickly troubleshoot all this. We appreciate your patience.
Can you hear me now?
Yes, we can hear you, Erin. Yay.
That's wonderful. Okay, I don't have the ability to turn my camera on because I am not a host or something now. I lost all of the controls for my Zoom. So that was the reason why I couldn't unmute myself. I apologize, everybody.
Well, let's go ahead and continue, Erin. If you can, are you able to move your slides back and forth?
Yes, that's a great question. It looks like things are moving.
Yeah.
All right.
Okay,
yes,
I'm going to take myself off camera, and it's all you now.
Okay, and we'll see if I can figure out how to. Yeah, I'm unable to turn my camera on, but I'm hopeful that I can see you during the Q&A. But for now, let me talk through and present some cool data from the NanoLive. So I'm going to talk to you about a commercial platform that EMSL has on site called the NanoLive. They offer several different platforms. The one that we have is the 3D Cell Explorer CXA platform, and its specialty is holography, and it is also able to collect fluorescent data. It is particularly strong in that it is able to image parallel samples and up to 60 replicates or different conditions at the same time. Let's see if I can advance this. All right, so let me give you a rundown of some of its capabilities. It has 2 different formats currently available, which is from one is from a 96 well structure that you image the inner 60 wells. That is a volume of between 80 and 90 microliters for each sample. The other format is a 4-well dish format that contains between 1 and 2 milliliters of liquid with your sample in it. There are also FITSI, TRITSI, and DAPI LED set for fluorescence at different wavelengths. The way that the platform works is that it has a rotating arm that collects spatial information for tomography from above. It also has a platform that is lidded and temperature controlled with a water reservoir to prevent evaporation from your samples. And you can manually designate the refractive index of any media that you choose. And so it's very flexible when it comes to choosing different media types or anything that's needed specifically for a novel or your favorite biological system. So because it's designed for time lapse, you do see that as you increase the surface area that you're imaging, you will have longer acquisition times. And this is particularly relevant to the well dish format, which can image up to a 10 by 10, what's called a grid scan surface area. So this will be in the very center of the dish. You're also able to navigate around the dish to choose what you would like to image prior to the beginning of your acquisition. So they've incorporated a number of different, very flexible, nice features in their software fairly recently that allows you more choice and flexibility. So I'm going to show you some examples of some data. I have several different organisms that I have images from. Because this is new, this is a new capability. This is not yet associated with any user projects, and so this was supported by EMSL leadership and some core funding in order to be able to collect this data. So I will take you through images of Nanochloropsis, which is a microalgae. It is a wild type, but it is contained and coexists with a bacterial flora, which you'll see. I'm also going to show you images of Euroia lipolytica, which is a yeast that we use as an oleaginous and bioengineering platform. And I will also show you images of Neurospora crassa, my favorite filamentous fungus. And there are also possibilities for different cell suspensions, communities, other kinds of samples. So I would love to have the opportunity to discuss any ideas that you have about imaging live cells. Let's see, trying to advance. There we go. Okay. Advantages of the NanoLive is that you can choose how long you would like to collect time-lapse data, and you can do so in a parallel fashion. So you can capture replicate wells in multiple conditions, which is really wonderful instead of having to do serial experiments. This is a platform designed specifically for label-free structural imaging. The other thing that the platform offers is a set of software that allows you to digitally stain your holography data. So you can take the densiometry that is collected and stain for specific densities. So you can image and then show different parts of the cell density associated with different organelles, for instance. There are multiple LED excitation wavelengths, and this decreases. We also have a fairly short light exposure during the acquisition that decreases phototoxicity compared to laser scanning confocal microscopy that tends to have much longer dwell times and either kill your cells or kill your fluorophore. The fluorescence data, however, is in 2 dimensions as opposed to the holography data, which you'll see is in 3 dimensions. As I mentioned, this is great for parallel experimentation. You can have many conditions, you can have multiple strains, you can image these in parallel so that you can do an experiment all at once without being concerned that you're collecting that the time is going to be a factor when you're doing your comparisons. So the resolution for each capture is approximately 100 micrometers on each side of a grid scan. So you can include a scale bar for that. This is just a subsection of a grid scan, and I have provided 2 different data types here for you. So this bottom image set here on the left is a series of germinating spore images of Neurospora crassa, and you have 2 different data types. The bottom is the holography data, and the top is a FITC channel that collects a GFP tagged protein that, and I made this strain. So the protein is a sphingolipid methyltransferase that is involved in a number of different processes, but it has a fusion to GFP, and you can see that the protein distributes itself throughout the cell, and the platform allows you to see those densities of the protein distribution. And at the same time, you're able to collect data like moving organelles. You can see structures where you have anastomosis or fusion tubes between these 2 different spores. You can see a whole bunch of different interesting cell biological phenomenon of the exact same cells over time. And then you also can do something like a 3-dimensional reconstruction of a cell in space because we have 3-dimensional data from the tomography. Let's see if I can advance this. All right. So some potential research applications that come to mind for me, but I would love to hear about any ideas that you have, would be something like lipid accumulation in oleaginous species, cell component analysis, so you can look and track the movement through of organelles or materials through fungal hyphae. You can do volume quantification of cell biomass. You can look at multiple organism interactions, and I'll have a couple of examples. There are also possibilities for morphological studies in response to nutrients, because you can do things in parallel with different media types. There are also, so you can look at simultaneous comparison of substrate usage in different strains to look at phenotypes or stress responses. And this platform is also great for stain and fluorescent probe imaging. So what you see here is another time series that is of the exact same cell that has another protein that has been GFP tagged that I made. So ACC1 is an enzyme that's involved in lipid biosynthesis, and it locates to the lipid droplet. So you can see as this cell goes through cell division, how this organelle is moving and how the organelles then become distributed to the new daughter cells. And we have then different data types that can be laid upon one another. It makes for some pretty cool figures here. So this first example is one of interaction between Nanochloropsis and its bacterial flora. So this was just in a still dish, so that you can see how Nanochloropsis settles and also how its bacterial flora changes over about 8 hours. We can look at a number of different cell features inside these cells, as well as look at how they change over time. Right. Okay. Another example I have again is Euroia. This is again the ACC1 tagged strain. This was captured in the 4-well dish format. And this was a single grid from a multiple grid capture that I did. And it is an overlay of both GFP, the FITSI channel with the holography data. But you can see about 16 hours of imaging. And this is the first section of it. What you'll see is one of the challenges that I'm going to highlight later, which is that the platform is best suited to thin layer samples. It was originally designed for adherent mammalian cells. And so when you have a significant height, or if you have things that would drift across the plane of image capture, that complicates the data collection. So things went a little bit out of focus late in the image, but some really interesting dynamics are available. This is another example where you can look at organism interactions or potentially interactions of different experimental conditions. In this case, I was attempting to compare, whether a treatment of the dish that I was using would affect the interaction of the species I was looking at. And so I was using polylysine in order to stick the cells to the bottom of the dish, and it did seem to have an effect. So what I see is that in the dish without polylysine, I have cell division and filamentation of Euroia, whereas in the dish that did have polylysine, in the presence of Pseudomonas, we had very little cell division occurring. So it looked like polylysine had an inhibitory effect on that particular set of organisms. Here is another example of organism interaction where we're looking at a Neurospora crassa spore suspension. So Neurospora is a filamentous fungus. So when then I mixed it with this Pseudomonas fluorescens strain, what I ended up seeing was there was less filamentation, there was less spore germination compared to the condition that did not have the bacteria present. So what kinds of data processing are available? So I mentioned that there is a software platform associated with the NanoLive. This is called Steve, and it is particularly designed for something that's called digital staining that allows you to take the density data coming from the tomography and false color it. I don't have an example of that. I'm happy to work with you if that's something that sounds like it would be interesting. The platform will allow you to make quantitative measurements, and it also provides stitched grid scan images. So instead of looking at single grid scans, you can look at a set of up to 10 by 10 grid scan images. The raw data is also compatible with ImageJ and Fiji. You get TIFF stacks or other types of export data that can be available both across time, but you can also get Z slices for tomography. So you can have slices at each time point to look at how the volume of your sample is changing and the density. You also then have a separate image series that is captured for fluorescence. And what you see here is an example of two different data types. One is the Z stack of the holography data, and the other is a TRITC channel, which was for a tracker for ceramide that is an interesting lipid that was related to some biological questions that I had. And this is false colored yellow. And so you have these overlayable different data types that are great for answering questions. So considerations and limitations to this platform. Imaging occurs from both above, and then light goes through the platform and is collected below. As a result, you need thin samples. You have also a 1-hour lead time that is required as the sample sits on the instrument, because the temperature of both the media and the cells have to come to an equilibrium in order to not have changes in focus during the acquisition. So focus does fail, and sometimes calibration of the instrument will fail with significant vertical height of the samples on the dish or the well. So that is a challenge if your samples have a particular shape to them, but it is navigable. So, there's some possibility for getting around that. If you have extensive cell growth that may, again, disrupt that Z-plane, that can be a challenge. Pre-cultivation for any samples is recommended for on-site live cell imaging. So we would want to have samples to grow on site. And we also prefer to start with a very low OD that makes the imaging and the thin layer of samples much easier to maintain. So additional information for the NanoLive capability is available on EMSL's Science tab, and I think that there may be links being dropped in the chat. You can find information under the Instruments and Resources tab under Optical Imaging. You can also include NanoLive on a letter of intent or in a proposal for capability requests. And please contact me if you have any questions or would like additional information about the NanoLive. Thank you. And let's turn it over to Dehong.
Okay, so I will continue for the second part of the talk. So it's about Lattice light sheet microscope. Basically, it's a fluorescence microscope. Okay, so once we talk about a fluorescence microscope for 3D imaging, you may think about confocal microscope. Confocal is a very popular. Maybe you have one you can access to in your institute. And of course, confocal is a very good. It's a easy to use. Then the confocal microscope, including many other microscopes, use basic configuration as shown in the left figure. Basically, the laser excitation light and the fluorescent emission light propagate to the same axis. So this way means everything along the laser propagation path get the light. Although you only image one focal plane, everything above and below all receive the laser illumination. That way cause some unnecessary photo bleaching and phototoxicity, even produce higher background. For this reason, Nobel Prize winner Dr. Bassett doesn't like confocal microscope. He wants a high speed, high resolution, low impact to image deep inside biological systems. So he invented the lattice light sheet microscope. The lattice light sheet microscope uses a different path. Basically, the excitation light and the fluorescent emission light perpendicular to each other. So this way use the excitation light very efficient. In the other word, only the place we want to see receive the excitation light. And the place we don't want to see do not receive the laser excitation. So this way, the phototoxicity and photo bleaching is low. So what's the advantage of the lattice light sheet microscope? As we say, okay, it has a low phototoxicity, low photo bleaching, and then low fluorescent background. Other advantages include the sensitivity to see single molecules. That's the good thing because later we can do single molecule localization for super resolution, also known as storm and PAR. Also, it uses camera to take one image, so the speed is faster than laser scanning confocal. Also, it offers isotropic 3D resolution, means X and Z resolution are similar, not like the confocal microscope. The X-ray resolution good, but the Z resolution is worse. This is important for 3D biological imaging because the biological sample usually is a 3D structure, also not oriented the way we want to the biological system to sit. There's no preferential axis. This axis we can get high resolution, but the other axis we don't care. So here I will show you some examples of what we have done. In the literature, you will see many beautiful images of a lattice light sheet of mammalian cells, but here I will show you those systems. I try to limit the system to those DOE relevant biological systems. So the first one is diatom. So I did confocal imaging. Diatom has a chlorophyll, so it has a red fluorescence. Also, we stain it with a Bredeweg dye to see the lipid droplet. The left side is a 3D confocal image. And then we did the 3D Z-stack of the confocal, and then I plot the cross-section view, so including the XZ plane and YZ plane. And then you can see from the Z-axis, the resolution is not that good. We cannot see clearly the fine structure of the chlorophyll. For the same system, we did on the lattice light sheet microscope. The left-hand image is the cross-section view. From there, you can see the fine structure of the chlorophyll. So you see better resolution. Also, you can see XY plane, and then the YZ plane has a similar resolution. So this is really nice for 3D imaging. And then for the same data set, I can plot the using 3D rotation view. So this is much better than the confocal microscope. For the confocal image, I don't want to show you the 3D rotation view since the Z resolution is poor. So you see, this is an example of to do 3D biological imaging. The lattice light sheet microscope is better than the confocal microscope. Here is another example. The cell is yeast Yerovia strain FEB 157. This strain expresses GFP, green fluorescent protein, at a plasma membrane. But however, the expression level is pretty low, only very small amount. So the fluorescence is not very strong. And then, so we did the confocal imaging. And then, can we see the GFP? Yes. So we just increase the laser power. So the confocal microscope still has the sensitivity to see it, just with a stronger laser. So we got a 2D image as shown here. Then can we do a 3D imaging? Yes. So we can just do a Z-stack, okay? Still no problem. And then let's increase it to another dimension, time lapse. So I did imaging every 20 minutes, for a 3D stack. Then here is the problem. The live cells are no longer growing. So here is the problem for 3D, for live cell imaging. Because of the phototoxicity, the cells got some problem. Then for the same system, we tried the lattice light sheet microscope. This one is much better. Then the left image is a 3D view of the cell. Then the right-hand side is a time lapse of a cross-section view. So the data actually has 4 dimensions, X, Y, and Z, and the time. So here I just use the cross-section to show the time lapse. So every 15 minutes, I took a 3D stack. This in for 2 hours. So you can see under the microscope, the yeast cells are still growing. So this thing demonstrates the lattice light sheet microscope has a lower phototoxicity. This is good for live cell imaging. It's much better than the confocal. So now I will show you some real research project using the lattice light sheet microscope. Then this one is EMSL project from Professor Doty, University of Washington. She's interested in how poplar tree get nitrogen as a nutrient. Bacterial interface play an important role for the poplar tree to get a nitrogen source. So this is a big project. And then here I only show one lattice light sheet image. So the system is a poplar tree grow with a special strain of bacteria endophytes. This bacteria express GFP, green fluorescent protein, while fixing nitrogen. So we grow this system and then put the poplar tree root under the microscope. The left is a 3D view. So you can see the bacteria as a little dot here, and then the plant tree root cell wall. And then the right-hand side is a cross-section view. So from the cross-section view, you really can see those bacteria are inside the root cells. And then also clearly see where they are. So this work just got published actually last week. This will show up on the journal website. So the link is here. Here is the second project. This one is from Professor Lu, University of Texas. He is interested in develop sensors for small molecules. So we know many small molecules like metal ions, like magnesium ion, and then organic molecules like ATP and glucose are very important in cell metabolism. But those things is very hard to probe under microscope. Unlike DNA, DNA is very easy. You can buy some commercial available dye, stain it, and then image it. But for those metal ions and organic small molecules, there's very few tools available. So he is developing sensors for this kind of small molecules. So this sensor has 3 domains. The first part is a DNA enzyme. So illustrate on the left side of the PowerPoint. Then the second part is a DNA paint. So illustrate on the right-hand side. The third part is antibody to anchor at a specific location. So DNA enzyme is a small DNA sequence which can sense and then bind specific molecules or metal ions. To obtain such a thing, Professor Lu's group begin with a random library, very large library, like 10 to the 15th sequences, then go through multiple iterations of selection and application, eventually obtain something working really good. Even it can even distinguish between iron 2+ and iron 3+. So the second part is for fluorescence detection. It's called DNA paint. It's a name is called DNA-based point accumulation for imaging in nanoscale topography. This one is published a few years ago by other research group. Basically, this method uses a small DNA oligomer labeled with fluorescent dye. The sequence is a complement to the sensor. So this one can transiently bind to the sensor and then diffuse away. So it has a fluorescent dye, so it can do fluorescent imaging. And then the nice thing about DNA paint is it's easy to multiplex. We can use different dye and different sequence for different sensor. The next thing is it's good for single molecule super resolution imaging like a storm, because this DNA paint image just transiently binded unbound. So it's equivalent to photo blinking of single molecules. However, this DNA paint has an issue is there's a free unbound fluorophore in the solution. It's just equivalent to immerse your cell into dye solution, so the background is high. Other people use turf microscopy, basically do total internal reflection, only image the one slice at the glass surface. But then here we will use the lattice light sheet to image the whole cell. The third part of the sensor is an antibody. So this thing can anchor on at the cell's specific location, so we can report the small molecules concentration at that specific location. For the demonstration purpose, here we just use microtubules antibody. So the location is a microtubule. So based on this way, we can do whole cells super resolution imaging. And then if you search this word 3D storm from literature, you will see many beautiful images. But however, those images usually is limited to something like a 1 micron range. Within the range, it shows the height of the object in very high resolution, including in Z. But you may ask, can 3D storm just do it like confocal, run Z-stack, just keep going up and down, then we build a whole volume? The problem of a 3D storm is you image one plane and then above, below, all those molecules get photo bleach. So it's very hard to do the next Z-stack, next Z-stack. Then this is the advantage for the lattice light sheet microscope. This one, since the excitation is a one thin layer, just as shown here. So this way we can just do storm slice by slice, and then every slice we can get a super resolution image, eventually build up the whole volume of the cell. So this is the right-hand side is a super resolution image of this DNA sensor to sense the magnesium ion at a location of microtubules. So with the lattice light sheet microscope, we can image the whole cell's volume with a super resolution. Then the third project here I'm going to show is from Professor Stacy, University of Missouri. He's interested in plant cell membrane receptors. That's hundreds of receptors at the plant cell membrane for different functions. So he asked the question, are those receptors in some nano domains? What are the interactions of different receptors? Do they form dimers, colocalize, or stay at the same nano domains or different nano domains? As this question, we need to use super resolution microscopy like STORM to answer those questions, because nano domains are pretty small. It's in a nano range, so we need a very high spatial resolution. Then before the lattice light sheet is available, we have to use a more conventional storm microscope. However, there's a issue of the autofluorescence from chlorophyll, just as illustrated on the right-hand side. We want to image the membrane, but there's a big chlorophyll on the top, produce a huge background. Even we choose the wavelength range with weak chlorophyll fluorescence, but it is still very strong compared to single molecules. So we couldn't do the work of a plant leaf. So we have to do it at root hair. So this is an image of a super resolution image of a root hair. So we label those plant receptors with antibody. For example, FS2 is a flagella receptor. P2K1 is a ATP receptor, and then SERK1 is a chitin receptor. All those different functions, then we can track in the single molecule level. And then we can do data analysis and analyze the distance. Since the talk here is about a microscope, I'm not going to go through those complex mathematical analysis. Basically, here I just present a solution. We see those receptors are with some nanodomains, but that may be at distinct nanodomains. Also, we observe a little bit co-localization between FS2 and P2K1, but little coordination between SERK1 and FS2. So this work was recently published. Now we have the lattice light sheet microscope. So we can really to study the plant leaf. The beauty of the lattice light sheet microscope is the excitation is from the side. So there's no big chlorophyll fluorescent at in the back. So it's very clear, and then there's no background. So here I just show you a 3D image. It's not super resolution yet. So just give you a sense of how the plant leaf look like under 3D view. So the left is a cross-section view. The right-hand side is a 3D rotation view. The green color is remorin. So it's the nanodomain marker with yellow fluorescent protein, and then the red color is chlorophyll autofluorescence. So we can see the cell membrane pretty clear, even with chlorophyll. So now we can do single molecule imaging to track those receptors. So this is a illustration. The left upper image is a 3D raw data image, and then the right-hand side is those tracked receptors, single molecule receptors. Then from there, we can calculate the distance. Then the analysis is still ongoing. So far, I show you those 3 real research projects, and then you may ask how the lattice light sheet microscope sample look like. What's the preparation of the sample? So basically, the sample is immersed inside aqueous solution, such as water, buffers, or growth media for live cell. And then for the literature and the microscope manufacturer, they usually suggest 5 millimeter round cover slip, and then put adhesion mammalian cells or even embryos on the top. Since we are DOE lab interested in other kind of systems. So we have done microbes and plant roots and leaves. And then for the sample preparation, I suggest you to contact me before you prepare the sample. If you put something at a microscope slide, like those tissue on the microscope slide, that thing will not work. So I suggest you to contact us before preparing the sample. And then for the more information of the instrument and how to request to use the instrument, those information is on the EMSL website, and then the link is posted here. Eventually, I want to thank the EMSL leadership to support this instrument, this technology, and promote this technology. For this 3 research project, I leave the PI professor's photo here. I also list those people actually made a sample for those images, or actually acquired the images. So here, so I also list those people here. Okay, finally, questions.
All right, thank you, Dehong. Thank you, Erin. Erin, you should be able to turn your camera on now. And success. Genoa, if you can go ahead and turn off the slideshow so that we can have our Q&A. I want to thank everyone for your patience with us. We've recently switched Zoom accounts and basically lost all of our defaults from what we were using before. So even though we've practiced, I'm sure you can understand, everyone, that there's growth and challenges, and so we will be able to continue getting better and better at these webinars in the future. So anyway, want to move on to the Q&A. So we've got quite a few audience questions for you too, and so let's just jump right in. I hope I'm pronouncing her name right. Marina asks, can we use, and I believe this is for NanoLive, can we use NanoLive on mammalian cells?
Absolutely. If you have a user project that is chosen that involves mammalian cells, that is what the microscope is designed for originally, especially if they're adherent and you can make a thin layer. It is great at imaging mammalian cells of that size and shape.
Yeah, okay. And let's see, for Dehong, we have a question for you. Erica asks, do you see future applications for these using the lattice light sheet techniques in human and animal cellular biology?
That's a lot. Basically, lots of examples in human and animal cells, but since EMSL, we try to focus on other things like bacteria and plants.
All right. Can you talk a little bit more about the advantages of NanoLive over standard confocal microscopy?
The major advantages that it has is parallel sample and data collection. So you can look at multiple replicates or parallel strains or sample conditions, and it's designed very specifically for time lapse for long periods of time with low phototoxicity. So those are the major reasons that you would want to use the NanoLive. I also wanted to chip in, if the NanoLive technique were being applied to human and animal cellular biology, I think one of the major ways that it would thrive is that you have the ability to look at densiometry. This is also true for the lattice light sheet, where you can look at cells without damaging them. So you have the ability to look at stain-free data, and that gives us the opportunity to not only look at cellular processes, but also cell interactions, either within a tissue or in a community, if you have mixtures of bacteria or eukaryotic cells or both.
Fantastic. All right. So let's see. Austin is asking, is there a wide range of compatible plates? My concern is that obligate aerobes will settle over the over the course of time, which can certainly affect the imaged phenomena.
That's true. So the requirement for the well dishes, which are the larger sized containers, is that they have slightly lower edges. That's to make sure that you have the ability to capture the tomography data by the rotating arm. The layer of liquid that is in it is pretty thin. So I do understand, though, the concern about oxygenation. The other interesting thing about the stage is that it is controlled, and you can pipe in CO2, but we could also potentially pipe in additional oxygen to enrich the atmosphere if you have an aerobe that has high oxygen requirements.
Mm-hmm. Okay, and then Sharon Doty, who was featured in our webinar today, she asks, most of your examples were eukaryotic. Can we look at the interaction of two bacterial species?
Absolutely. That's not, I was looking at a particular Pseudomonas fluorescens strain that I had that has an MK tag that was easy to look at to compare red and green channels. But bacterial species in communities are definitely within scope. And yes, it would be easy to look at physical interactions, and you would get spatial information looking at any kinds of bacterial interactions.
All right. Okay, so we've got, let's see, for Dehong. So Marina is asking too, for the Lattice light sheet, is it possible to use FFPE fixed stained human tissue sections?
We never try. Then the thing is, those fixed tissue sections usually put on the microscope slide. Then the standard microscope slide, like a 1 inch by 3 inch size, is not compatible with the microscope. So those tissue section needs to be directly put on 5 millimeter cover slip. That's a pretty hard thing.
Mm-hmm. Okay, so, and I apologize in advance, I hope I'm saying their name correctly, but Marichelle?
Marichelle.
Marichelle, there we go.
I know
Almost
Marichelle.
there.
Yes. Hi, Marichelle.
Yes, in fact, Marichelle says hi as well. Great to see you. Would like to know, what is the maximum resolution for NanoLive? Could we look at organelle dynamics during Candida germination? That's one question for you, Erin. And then I believe there's another question for you, Dehong, but let's, Erin, if you can answer that one, then we'll get to Marichelle's other question.
So the smallest imaging area that you can do is about 100 by 100 micrometers. That's one grid scan. But you are able to see individual organelles and their different densities, and it's compatible with either using that density to track the organelle dynamics, or if you have an organelle specific stain or a fluorescent tag that you want to use, that's also compatible with any kinds of questions that you have during germination. I will let Dehong talk about the Lattice light sheet.
You're right. So her question for Lattice light sheet, have you tried using Lattice light sheet for Neurospora? Could we run some trial runs before writing a proposal?
I haven't tried it before, but I think it's possible to do it. It's possible to try. Yeah.
Yeah, and I have Neurospora on site. We could coordinate something just to see what we could see.
Great. So we've got 3 minutes left, so probably a time for one more question. Oh, and by the way, Marichelle says, thank you, I will email you. And let's see for how, so let's talk about the advantages of the Lattice light sheet over other microscopy. Dehong, do you have maybe just one or two quick, blurbs about the advantages of it over other microscopy?
It's a low phototoxicity, low photo bleaching, and then the isotropic 3D resolution. I think that's the most important thing, distinguished from other microscopes.
Okay. All right. Well, I'm not seeing any more questions from the audience. And so last call, if anybody has a question, otherwise we can, okay. I'm not seeing any more questions. So we can wrap up this webinar. Thank you everyone again for your attendance. And don't forget, on February 7th, we have our next webinar on metabolomics technologies at EMSL. So we hope to see you there. So have a great rest of your Wednesday. Happy New Year.
Please follow up with any questions by email if you have any. Happy to chat.
Absolutely. Absolutely.